My name is John Hopper. I am the chairman of the Hopper-Belmont Foundation, and we are so glad you're joining us. What we hope will be the first of many presentations from the amazing early career cancer researchers who have won the Foundation's Inspiration Award due to their exceptional studies on early detection, early intervention, and curative therapies in the areas of pancreatic, ovarian, and pediatric brain cancers. Thanks to so many of you, by the end of 2025, the Foundation will have recognized at least 25 early career MDs and PhDs out of over 120 applications. Today's presenters were chosen from among these alumni for outstanding research publications in their area of expertise, in this case, pancreatic cancer, thanks to a new annual Career Development Award supported by Mr. Frank Cowan. Some quick production notes for everyone. With the exception of the presenters, all of you logged in will have camera and audio turned off by a master switch, but we know that you're here. During the presentations, we encourage you to interface with questions via the Q&A tab located at the bottom right of your screen where you see 9 dots. So if you tap that on, and I think if you scroll down, you'll see something that says Q&A, and we encourage everybody during the presentations to ask questions. The way it will work is our presenters tonight will have 10 minutes each with about 5 minutes afterwards for Q&A, and with any time remaining, we'll open up to all questions to anyone. So let me introduce our presenters tonight. They include Dr. Anupriya Singhal from Memorial Sloan Kettering, Dr. Jackie Zimmerman from Johns Hopkins, and Dr. Willian Hwang from Harvard Mass General. And again, each of these great folks will have 10 minutes to share their great research. And let's begin. So Dr. Singhal, the stage is yours for the next 10 minutes.
All right, well, thanks so much for the opportunity to present. I'll just pull up my slides. Okay, hopefully this looks okay on your end. So just by way of introduction, I'm junior faculty on the GI Oncology service at Memorial Sloan Kettering, and I'm also a lab researcher. So I do lab research the majority of the time, and then I also have a clinical practice focused in treating patients with pancreatic cancer and other GI cancers. When I started my work in the lab a few years ago, I was really interested in this question of how heterogeneity within tumors drives drug resistance. And within the lab, I've been thinking primarily about transcriptional heterogeneity, which is this idea that cancers can actually often arise in a fixed genetic context, but cells often differ from each other considerably in terms of their transcriptional profile or cell state. And so in the context of diverse cell states, I wanted to ask some basic questions in cancer evolution. So first, how do therapies select for certain cell states but eliminate others? And importantly, what are the cells that actually compose residual disease after treatment? And clinically, the importance of these cells, or these are the cells that are anticipated to give rise to relapse. And then I also wanted to ask, how does cellular plasticity or cell phenotype switching occur during tumor development? And is this a way that cancer cells evade treatment? So we've been thinking about these questions in the context of pancreatic cancer, and in particular in the context of new drugs that are coming to the therapy of this particular disease. And as we know, KRAS is a dominant oncogene in pancreas cancer, with the vast majority of tumors having a mutation within this gene. And excitingly, there's a whole suite of small molecule inhibitors of KRAS that are also coming now into the clinic. And so understanding resistance mechanisms to these drugs is of strong clinical importance. I think excitingly, there is evidence that, these drugs have activity in pancreas cancer. And on the left, you're seeing a small study from patients with pancreas cancer that have a G12C mutation that then went on to receive treatment with the KRAS G12C inhibitor. And what's been exciting to see is that the disease control rate is actually quite high for these drugs, so upwards of 80%. But I think one disappointment has been that the durability of response still remains relatively short. So here, 4 months in this study. And so what many groups have started to do is to look at mechanisms of acquired resistance. So really understanding what is the genetic profile of tumors that manage to resist and then relapse on therapy. And, one emerging theme is that many tumors actually appear to have divergent mechanisms of genetic resistance. So on the left, you're seeing patients that had pre-treatment and post-treatment cell-free DNA taken. And what you can see is that in individual patients, unfortunately, oftentimes we're seeing multiple mechanisms of genetic resistance that allow for relapse to these drugs. And so I think the takeaway point from this is, unfortunately, it may be the case that it's quite hard to sequence therapies to combat acquired resistance to this drug class. And so what this led us to do is to start looking in the lab early on to see what are the features of residual disease after treatment, and might looking at features of residual disease actually afford us an opportunity to intervene earlier on in treatment and then eliminate these particular cancer cells. So in pancreas cancer, we're fortunate that transcriptional heterogeneity prior to treatment has actually been relatively well defined. And, studies over the last 10 to 15 years have shown us that in the majority of human tumors, there are both classical cancer states and basal cancer states, and that these states coexist within individual tumors. And you can see this nicely on the image on the lower left. Classical cancer states tend to be associated with well-differentiated histology and also better prognosis in patients, and basal cancer states tend to be associated with aggressive biology and are generally more therapy resistant. And so we wanted to ask, again, in the context of these diverse cell states, which are the cell populations that are actually acutely resistant to RAS inhibitors? And so this is part of the study that was published last year. And what we did was we took the genetically engineered mouse models of pancreatic cancer, where we know pre-treatment tumors actually do harbor this classical basal heterogeneity. And then we treated with RAS inhibitors. And what you can see is that residual disease, sort of surprisingly, actually has very classical or epithelial features. You can see that many of these cells look very glandular under the microscope, much more so than the pre-treatment tumors. And so these are the cells that are actually resisting therapy. We looked at this in a variety of different modalities, so not only by histological analysis, but also by single-cell RNA sequencing. We saw this very strong shift towards a classical identity under treatment. To really be sure that this was not just a feature of the mouse model, but something that pertains to human tumors as well, we took a series of xenograft models, all of which were KRAS G12D mutant, but also had a variety of other genetic alterations, and then again treated with MRTX1133, the KRAS G12D inhibitor. And what we saw, again, was that cells that are acutely resistant to this drug class always seem to have these classical features. So again, very epithelial under the microscope, and oftentimes containing intracellular mucin, really suggesting their epithelial identity. So this was quite interesting to us, but we wanted to also take this one step further and see if we could actually prove that these classical cells that survive therapy are functionally important, so that they give rise to relapse disease, and then also that if we were to eliminate these cells alongside RAS inhibition, we would be able to improve depth or durability of response. So in order to address these questions, we developed a new genetic model that has kind of a lot of bells and whistles, but essentially it's a model that allows us to really functionally interrogate this particular cell population. And this is a genetic construct that we generated that has multiple different components, but for the purpose of this talk, I'll focus on our ability to lineage trace classical cancer cells. So what lineage tracing in this context means is that you can introduce a heritable genetic mark within classical cells and then ask what's happening to those cells upon relapse. And then you also have the ability within this system to ablate that cell population selectively. So we can basically just kill off the classical cells and then ask what's happening to the tumor as a whole. As you can get a sense of, this entire genetic construct is integrated into this classical gene, galectin-4, and this is what transcriptionally restricts the expression of all these different components into the classical cells. When we generate tumors that are made from this cell line, you can see that this reporter system actually is quite a faithful model for the classical cells. And the classical cells are those that are red here and very epithelial appearing under the microscope. And these are the ones that are expressing the reporter system. So using the system, we first asked this question, what is happening to the classical cells after KRAS inhibition? So we first treat the tumors down to residual disease time point, and then we introduce this heritable genetic mark, which is blue fluorescent protein. And you can see on the left that this mark is actually very selective. So only these residual classical cells are those that end up getting this heritable mark. And then we ask, what happens to these cells over time? So at the time point of acquired relapse, we were excited to see that not only do classical cells give rise to these more aggressive looking cells at the time of relapse, but also that these blue fluorescent cells, again, derived from classical cells, start to make up a large fraction of the tumor, really proving that classical cells do give rise to relapse. So next we wanted to ask, what's the therapeutic benefit of eliminating this cell population? We did this again using our reporter system, and here we can use the ability of our reporter system to, we selectively eliminate the classical cells using diphtheria toxin. And, what we see is that, RAS inhibition has a very strong effect in terms of overall tumor burden, but if we combine this with classical state elimination, we get an even stronger or deeper response in the tumors. And so, we also asked, are there kind of existing therapeutics that we can use alongside RAS inhibitor that would achieve a similar effect? And we know that clinically chemotherapy is actually, such a drug, or that classical tumors tend to benefit more so from chemotherapy. And so this provided a biological rationale for combining KRAS inhibition with chemotherapy in order to get, again, deeper and more durable responses. And so in our experiments on the right, you can see that combining RAS inhibitors with standard of care gemcitabine and nab-paclitaxel both increases depth of response at early time points and also increases the durability of response. And excitingly, these are combinations that are already in clinical trials, and some of our data really points to what the biological rationale is for combining these two drug classes. So as we... Okay, sounds good. Yeah, I'm just about there. So as we move forward, we're really thinking about now how do we bring this finding kind of translationally and into patient care. And what we've been starting to do is to look at features of residual disease after RAS inhibitor therapy in patients. In collaboration with our clinical colleagues and pathology colleagues, and also due to the generosity of patients that are willing to undergo pre-treatment and on-treatment biopsies, we've been looking at features of residual disease after KRAS inhibitor therapy and trying to identify proteins that are upregulated in this context that would allow us to, again, rationally combine existing therapies with RAS inhibitors to achieve similar effect. So with that, I'll just quickly summarize this work. What we found in this study is that the basal cancer state in pancreas cancer is highly sensitive to RAS inhibition. This is a phenotype we observe across mouse models and in patient-derived xenografts. And we're starting to see these features also emerge in human disease. And what we showed, again, using our model systems, is that these classical cells actually serve as a reservoir for relapse disease. So there's a very strong rationale for trying to identify combination strategies that eliminate this cell state alongside RAS inhibitors. So with that, I'll just quickly thank my mentor, Tomas Tamela, as well as the rest of the lab, and then my colleagues and mentors on the GI Oncology service at Memorial, and of course, our funding sources. With that, I can take any questions.
Thank you, Dr. Singhal. That was very interesting. You've actually answered a lot of the questions that came up during your presentation, but one question, is this phenomenon of classic cell resistance also seen in vitro or need vivo context?
Yeah, it's a great question. So, it's one that comes up a lot. I think what we've seen, and I think other groups have seen this as well, is that the in vitro context often does not recapitulate the diversity of cell states that we see in the in vivo context. So for example, most mouse in vivo tumors and human in vivo tumors have both classical and basal cells, whereas actually different in vitro conditions can support one cell state more so than another. And so we don't actually reliably see this classical state transition occurring in different in vitro systems, but it is something that we see sort of regardless of the in vivo context.
Thank you. I'll give people 1 more minute or to ask a question online. Otherwise,
Jill, I see one other question here, which is to Dr. Singhal. You'd mentioned clinical trials, I think, on this in terms of, if I think people heard correctly. So what exactly is the next step in terms of making this into bringing all this to translational therapies?
Yeah, so I think it's a great question. I mean, as we know, right, many of the drugs are still in relatively early phase testing, and I do think there's a little bit of there will be a little bit of a timeline before we're able to start working towards novel combination strategies. But as I sort of mentioned in the talk, there are already existing therapeutics beyond chemotherapy, which would be expected to kind of eliminate these classical cell states that are resistant to RAS inhibitors directly. Some of these are antibody drug conjugates, so basically, targeting abnormal proteins that are expressed specifically on classical cancer cells. Some of the targets that are already in development are targets like MUC1 or Claudin 18 that are already being tested in pancreas cancer and other contexts. And I think what this work that I described tells us is that there is a rational combination strategy that takes these antibody drug conjugates and combines them with RAS inhibitors where there's expected to see, benefit. So I'm hopeful that kind of our next wave of combination trials will include combinations beyond just chemotherapy and also potentially rational use of these types of drugs, like I mentioned, ADCs.
Great. Thank you so much, Dr. Singhal. Dr. Zimmerman, you are up next.
Thank you. I'm going to share my slides really quickly. Okay, so hopefully everyone can see my slides and their total. So my name is Jackie Zimmerman. I'm an assistant professor and a GI medical oncologist at Johns Hopkins. And I am going to briefly summarize our program today and then a subset of the findings that were part of my Inspiration Award, and then broadly discuss some of the next steps that are underway. So whereas Dr. Singhal gave us a beautiful overview of some of the tumor cell subtypes and the considerations of RAS inhibitors, I'm going to talk a little bit about untangling tumor cell and cancer-associated fibroblast interactions in pancreatic cancer. So our group is interested in the heterogeneity of pancreatic cancer, recognizing that while important, the tumor cells don't tell the whole story. This cartoon does a nice job representing the complexity of the tumor microenvironment. Cancer-associated fibroblasts, which have these spindle-like structures that you're able to see here, represent a large proportion of the tumor microenvironment and are increasingly appreciated for their complex contribution to tumor behavior. You can also see that there's a diverse network of immune cells as well within the PDAC tumor microenvironment, which I'll focus on a little bit less today. And then you can see highlighted along the bottom of this cartoon some of the different approaches, processes, and targets that have been used to evaluate the stroma or target the stroma from a therapeutic perspective. And overall, this has been a relatively disappointing effort, but I think an area where we have a lot of opportunity for improvement in the next several years. So our group uses a combination of patient-derived models and computational tools to understand cellular crosstalk in pancreatic cancer and identify candidate intercellular signaling that we can leverage therapeutically. So our institution has a very busy surgical practice, and we're able to readily obtain tumor tissue from patients at the time of surgical resection after they're consented on biobanking protocols. So briefly, once the patient goes to the OR, we get a piece of tissue and we basically fraction it out and use a portion of it to generate these patient-derived organoids or individual avatars to represent the patient's tumor, giving us an opportunity at capturing interpatient variability. And then given our interest in cancer-associated fibroblasts, or CAFs, we in parallel isolate and reconstitute the CAF fraction of the tumor microenvironment. We also have the capacity to reconstitute immune components, either from peripheral blood obtained at the time of surgery or from tumor-infiltrating lymphocytes. But again, that was less of the focus of this particular project. And then we're able to subsequently reconstitute a reductionist tumor microenvironment that we have control of for subsequent experiments and downstream questions. So for these multi-compartment avatars, we have a number of downstream applications. Tonight, what I'm going to highlight is the capacity to identify intercellular interactions, as well as the capacity to integrate the data from these models with some of our computational tools and predictive algorithms. So in real time, this is what this looks like in culture over about a 4-day period. You can see in the middle, the patient-derived organoid proliferating and has this sort of pseudoductal shape. And then going through our surrogate basement membrane, you have these spindle-like cancer-associated fibroblasts just making their way through the matrix. So since our organoids represent a relatively small number of patients, we complement this model system by applying computational tools to larger data sets to validate our conclusions. And this has also served as a way for us to benchmark our organoid system. So in this manuscript, we published a single-cell RNA sequencing atlas looking at the gene expression of 77 patients, 61 of whom had a diagnosis of pancreatic cancer, and then we had 16 non-cancer tissues. And so the figures here just demonstrate the overall cellular makeup of the tissue samples, as well as the samples and cells that were represented from each data set. So you can see that broadly, we were able to capture a wide array of cell types, including a strong representation of fibroblast populations as well as tumor cell populations. And so we termed this an atlas, and it has not only informed our work, but it's also something that's available to our colleagues who may have questions that could be answered by this resource. So we think of it as a shareable tool in addition to facilitating our own work. So we first applied a computational approach to the atlas to better infer tumor and CAF interactions. And this was an algorithm called Domino. And so the network diagram you can see on the left highlights the individual relationships with the line width and node size indicating the strength of the relationship between cell types. And so for this inference, we subset populations of tumor cells, as well as cancer-associated fibroblasts for additional granularity. But for today, I'm just going to focus on broadly the tumor cell and the CAF populations. And so using this approach, we identified a possible ligand and receptor relationship, or a crosstalk relationship, between tumor cells and CAFs that was mediated through VEGF-A. And so the heat map you can see on the right highlights this relationship, demonstrating an incoming signal from the tumor cells to one of the CAF populations. We saw that this relationship also held up across other CAF populations as well, and we were particularly intrigued by this relationship since there's already FDA-approved agents targeting VEGF-A, most notably bevacizumab, which goes by the trade name Avastin. And so we took this finding back into our individual patient organoids using CAF and organoid co-culture to see if this dynamic relationship held true. And since our atlas was looking at gene expression or RNA levels, we first used qPCR to see if we could recapitulate what we found in the larger patient atlas. And interestingly, we were able to demonstrate that, in fact, there was more VEGF-A RNA in the co-culture relative to the monoculture counterpart, suggesting that expression was being triggered by the presence of the second cell type. Similarly, when we looked at the amount of VEGF protein that was being secreted from the co-culture, we saw that that exceeded what was being secreted from the monoculture. Again, being conscious of the fact that our system is very reductionist and does not comprise the entirety of the tumor microenvironment, we took this back to matched patient tissue and just validated using immunohistochemistry that there was, in fact, a strong VEGF presence in those tissue. And this wasn't an artifact simply of our system. To further try to understand the mechanism of VEGF signaling and kind of the sender receiver of this interaction, we looked for concurrent changes in surface expression of the VEGF receptor 1 and 2, which are the most commonly seen VEGF receptors, and found that, interestingly, there were no concurrent changes in the receptor expression in the co-culture as compared to the monoculture. So given the lack of changes, we went back to our domino inferences to look again and see if there was another potential receptor that we could identify. And so in doing so, we identified neuropilin-1 or NRP1 as a receptor associated with VEGF-A, and then reapplied this inference into the co-culture and found that indeed, the co-culture demonstrated a downregulation of neuropilin-1 expression on the surface of the cells, which was consistent with increased receptor binding and recycling in the co-culture in the setting of the increased VEGF expression. And so finally, we returned back to our reference tissue from which the organoids were derived to again verify and prove to ourselves that we did have co-localization of the neuropilin-1 receptor with VEGF alpha. And you can see from the map here that we had a correlation plot that showed that in fact spatially the presence of VEGF-A correlated with the presence of neuropilin-1, again kind of reinforcing that this relationship is one that holds true in a complete TME and not just our reductionist system. So in summary, the purpose of this project was to establish a pipeline for generating multi-compartmental biobanks and converging that with our computational tools. So in order to accomplish this, we created a single-cell RNA sequencing atlas from published data sets that we made shareable to our colleagues. We then generated a multi-compartmental patient-derived organoid biobank with organoids, calves, and then, as I mentioned, immune cells that we've saved for later. We then used our computational tools. The one that I highlighted today was the domino tool to guide inferences about how the cells interact with each other to guide underlying tumor biology. And then finally, we identified a relationship that is targetable and actionable between the tumor cells and the calves mediated through VEGF-A and neuropilin-1. And we hypothesized that this interaction plays a role in enhanced EMT and inflammation, which is something that we saw kind of subsequent to this capping point of the story. We further plan to use this particular workflow and framework to apply to future projects, including those with immune cells represented into our system. So broadly moving forward, our goal is to prioritize targetable interactions to evaluate for sequential treatment interventions and cellular reprogramming, to integrate immune cell populations to examine the role of the calves in contributing to the immunosuppressive tumor microenvironment, to ultimately allow us to evaluate mechanisms of immune activation and overcome the immunosuppressive tumor microenvironment. So with that, I'd like to acknowledge our lab, my mentors, and our funding sources, and absolutely the generosity of the patients who are so kind to share their tissue with us.
Thank you, Dr. Zimmerman. Joan, I have a question here that came up. Do you have one in your?
No, but if I can just interrupt, if anybody has any questions and they got on a little bit late, in the bottom right-hand corner of your screen, there are 9 dots.
Joan, you just mute again.
Sorry about that. If you go to the bottom right-hand corner of your screen, the 9 dots, scroll down to Q&A, and you can ask your question there. Sorry.
Okay. Okay, I think one of the questions I see here, Joan, is to Dr. What's the timeline going forward on this in terms of your next steps?
Yeah, that's a great question. So each of these models takes us between 5 and 8 weeks to generate. And so that is a little rate limiting step in terms of the preparation to move forward. Certainly, since we're banking everything, we have a few hundred models that are readily available to us that represent a variety of genetic and treatment statuses of the patients who have been generous enough to consent on this protocol. Moving forward, we've actually already integrated T cells into the system, which allows us to look at the combination of both CAF modulation as well as immune modulation simultaneously or sequentially. We're in the process of integrating macrophages presently. And I think, what's nice about this platform, which is very labor intensive to get set up, is that once it's set up, it is a glorified in vitro system, which lends itself to relatively rapid assays in terms of drug screening and relatively rapid functional assays, as well as we're starting to try to better understand underlying mechanism.
Great. I think we have time for one more before we get Dr. Hwang on there. Joe, I see the one.
Yep. Given that the drugs targeting VEGF-A already exist, what are the specific considerations or challenges in repurposing or applying these existing drugs based on your findings, especially considering the role of neuropilin-1?
Yeah, it's a great question. I think it's challenging because this particular mechanism has been somewhat underwhelming in pancreatic cancer. And I think what we're thinking about more and more as we think about the complexity of the microenvironment is that the process is going to be one of reprogramming and sequential modulation. And we're not necessarily going to have a single mechanism that we can target with any sort of definitive and durable response, similar to what Dr. Singhal was alluding to in combining the chemotherapy with the RAS inhibitors. So I think the nice thing about the VEGF mechanism is that there is FDA approval for these agents already. So it's kind of already in our toolbox to use. The challenging thing is getting enthusiasm for it because it has performed in an underwhelming fashion in GI cancers in general. And I think that that there is a lot of excitement to think about it biologically and again in combination with other agents, and that's what we're exploring right now.
Gotcha. Yeah.
I think we have a couple minutes, John. So if we can just ask her, we've got a couple more questions that were asked. If you can answer them kind of quickly. How do the phenotypes in your co-culture systems map to those seen in patient samples atlases?
Yeah, that's a great question. And I think to Dr. Singhal's earlier point, we do lose some of the heterogeneity over time. It's a little bit of a balancing act. We've got to have enough biomass to be able to do our experiments with rigor, but we do sacrifice some of the heterogeneity. We are able to see fluctuations in the heterogeneity, particularly when we induce things with culture conditions, other cell types, conditioned media. So it does give us an opportunity to look at dynamics, but we certainly are sacrificing the comprehensive heterogeneity, which is why we've opted to partner the in vitro work with tissue-based assays and the patient single cell atlases.
Okay, and one last one. It says, really exciting model system and data, Jackie. In your co-culture system, do the CAFs maintain their heterogeneity or do they adopt a MyCAF state?
Yeah, that's a great question. They do tend to adopt more of a MyCAF transcriptional state and functional state when they hit plastic. When we start to culture them, we are able to pull back that phenotype a little bit, again, when we kind of modify the culture system to give us a little bit more representation of other fibroblast. I think, to really capture heterogeneity in a focused way, it does kind of force us to do some genetic modification of those cells to be really confident that we are reliably obtaining the phenotype that we hope to obtain.
Great. Thank you, Dr. Zimmerman, Dr. Singhal, Dr. Hwang. Really fantastic presentations, really great publications. I think it shows a lot of promise for pancreatic cancer. A big thanks to Joan Fowler here, who's our executive director and on the board, and John, J-O-N Hopper, for putting this together, and everyone else has been helping to get this as our first inaugural Career Development Award presentations. Hopefully we'll have many more of these coming forward. And again, thank you for everybody who came on board to ask some questions. This is, again, hopefully very informative, very promising, and gets us all energized for what can be in the future. So thank you, everybody. Appreciate it. We're right on time. Is there any last questions before we sign off? If not, thank you so much, and more to come. Take care.
Thank you.